I would like to welcome everybody to the National Beef Cattle Evaluation Consortium 2019 Brown Bagger Series. This is session 3. Hopefully you've been with us for the other 2, and we have one more to go next week that we'll talk about at the end of today's presentations. As always, I think by now you should know about our website. It's good for finding the archived old brown baggers, and we encourage you to go and utilize these, and the information that's shared on these are free for your usage and educational purposes as well. I would like to welcome you. My name is Darrh Bullock. I'm an extension professor at the University of Kentucky. I've been here for a couple of years now, and I'm one of 3 members of the Education Committee, myself and Bob Weber and Matt Spangler. Bob at Kansas State and Matt at the University of Nebraska are the coordinators of the educational program, which for the most part consists of this Brown Bagger series to date. Through the consortium, and today or this year we hope we've put together another good program for you guys. Participation has seemed to be very strong, and we do appreciate you guys. As long as you guys want the information, we're going to continue to try and provide these brown baggers for you each year in October. Today we have a couple of good speakers, great speakers, and good people as well. We have Dr. Alison VanEenennaam and Dr. Megan Rolf that will be our speakers today. I'm going to do things a little different. I'm going to introduce Alison and let her go, and then I'll introduce Megan before her talk while we're making the transition. But Alison is going to be providing us a presentation today on genome editing in beef cattle, recent developments and prospects. For those of you that don't know Alison, and I would find that hard to believe, but Alison comes to us from down under, way down south in Australia, where she got her bachelor's at the University of Melbourne, and then she came over to Davis, where she got her master's and her PhD, and currently is working as an extension specialist at UC Davis currently. Alison does, for those of you that may not know, she does an extreme amount for being an advocate for biotechnology, particularly within agriculture, and has always been very passionate about encouraging sound scientific information and making sure we make our decisions and base our decisions on sound science. And I think that her position kind of opened the eyes to a lot of people across the country, and the need that we have for people like Alison who really promote what we're doing and making sure that what we're doing is done the correct way. So, I thought I knew Alison as good as anybody, but when I got her bio, I found out there was one piece of information I didn't know. Alison was this year's recipient of the Animal Science, American Society of Animal Science Rockefeller Prentice Award in Animal Breeding and Genetics. For those of you that don't know, that's a big deal. And congratulations, Alison. And I'm going to turn it over to you and let you tell. Oh, I forgot. There's Alison's picture. Sorry. And so, Alison, I'm going to turn it over to you. If I can figure out, I'm going to stop sharing. And now you take control.
All right. Are you seeing my screen?
I sure am.
All right. Well, magic. Thanks for the introduction, Darrh. And I'm going to get right into this because there's a lot happening in this space, and gene editing has been in the news a bit recently. I'll talk a little bit about some of the reason it's been in the news, but you might also have seen there's a new Netflix series out called Unnatural Selection that talks about gene editing. I'm going to focus on it today as it relates to its use in livestock for breeding purposes, but I figure I better give a little overview of what gene editing is, and so we can be starting on the same page. So gene editing basically involves the use of these what are called nucleases. In other words, they're proteins that cut the double-stranded helix of DNA. And there's some that have been around about 20 years, zinc fingers and talons, but the one that most often you hear in the press is this CRISPR-Cas9. And basically all of them do the same thing, and that is they make a double-stranded break in the DNA at a particular genetic location that you can tell them to go cut at that particular DNA sequence. And given the A's and T's and G's and C's and the order of them, you can basically very precisely tell them to go cut on chromosome 1 or chromosome 10 or wherever it is that you would like them to cut. And once they've cut, in other words, they've induced a double-stranded break, shown here as a little lightning rod, the cells try to repair a double-stranded break because nature doesn't like double-stranded breaks. And they do it in one of two ways. They can go down what I call the left-hand pathway here, where very fancily they just stick the break back together and cross your fingers that you didn't mess up too badly. And sometimes you get maybe a single base pair or a couple of base pairs that drop out or get added in that repair. And that can result in a change in the reading frame of the codons that make up the proteins that make up the gene. In other words, you can break the gene so that that protein is no longer expressed, and that gives you what's called a knockout. And that's actually something that happens a lot in cells, and that's actually the basis of evolution, and all of our breeding programs is working on this type of natural repair mechanism called non-homology end joining. That's distinct and different from this other repair mechanism. I'm going to call this the right-hand side pathway, where you want to intentionally insert a piece of DNA between the break. And to do that, you provide it with donor template, shown here in pale blue. And the donor template has sequence homology. In other words, the sequence on either side of the break is the same in this donor. And when it goes through this homology-directed repair, you basically get an insertion of this piece of blue DNA. And that can be as simple as maybe including an allele that you would like in your breed. So maybe you'd like to bring the allele for polled into a breed that's got horns at that particular loci, or maybe you want to bring in an entirely different gene from even as far as a different species and go full transgenic. So there are two kind of repair pathways, and I want to focus a little bit on this homology-directed repair pathway because it's going to be relevant to what I talk about later. So here's CRISPR-Cas9, and we've got donor template, and you see the insertion or knock-in like I described on the previous slide. But you might wonder, well, how is this donor template supplied? Where does this come from? So typically when you're editing, you introduce the protein, CRISPR-Cas9 in this case, and then you will introduce the donor template sometimes on what's called a plasmid. And a plasmid is a circular piece of DNA that replicates in bacteria. And what geneticists do is they create recombinant DNA where this yellow sequence is what we would like to insert, and it's flanked on either side by homology arms, in other words, DNA sequence similarities to either side of our break here. And when this repairs via homology-directed repair, in other words, the right-hand side of the pathway, what we expect is to get the repair looking like the bottom here, where the yellow is the inserted sequence, and either side is basically just the same DNA that existed in that animal before we did the edit. And this vector backbone is kind of a molecular geeky term for the rest of the DNA that exists on this plasmid that enables it to grow in bacteria. It's not expected to be part of the repair at the end of the day. And you'll understand why I emphasize that as we go on in this brown bagger. So how might genome editing be used in breeding programs? Basically, it can be used to inactivate genes that encode for a protein that we don't want. So if a protein encodes and makes an animal susceptible to disease, then maybe we would like to go down that left-hand pathway and knock out that gene so it no longer makes that protein, so the animal's no longer susceptible to disease. Or maybe we want to introduce an allele from one breed I don't know, Angus, into, for example, Holstein, to give the no horns or polled trait. And that's an example I'll talk about in this presentation. That was done by a company called Recombinetics back in 2016. And basically what this list shows is potential targets, or actually reduced to practice targets, of different traits that you might use, target using genome editing. So you might want to introduce polled, you might want to introduce the centerpole mutation called SLIC, which gives animals heat tolerance. You might want to introduce that, for example, into dairy cattle. You might want to introduce a knockout of myostatin, which is what gives Belgian blue their double muscle characteristic. And so you could introduce that into a different breed like Nelore. You might want to eliminate a milk allergen like beta-lactoglobulin by inactivating that gene. You might want to eliminate the protein that's responsible for mad cow disease. You might want to alter the allele to be improved meat tenderness alleles for kelpane and kelpistatin. Or you might want to do homology-directed repair and introduce a gene that gives resistance to mastitis, for example, lyphostatin or lysozyme. Make an alteration that makes animals more resistant to bovine respiratory disease, resistance to tuberculosis, or some of the more unusual ones, perhaps introduce a gene that results in all of the offspring of an animal being male. Or perhaps you want to do something to make animals that are no longer fertile themselves, such that they can be surrogate sires for germ cell transfer from a different animal. And that's a really interesting area that I won't get into today, but I guess there's a lot of different potential applications that you could use this technology for. And here's one example, and that is genome editing to produce tuberculosis-resistant cattle. And this is work that's being done in China. There's a lot of work getting done in China. And basically addressing a zoonotic disease. Obviously, tuberculosis is human transmissible, and it's actually quite a problem in the UK where it's getting spread by badgers, ironically enough. And there's an interesting fight between the badger lovers and people that are not that keen on TB. So that's the type of thing you can use it for. And of course, as a geneticist, I say, what better approach to solving disease than genetic improvement, because it's permanent and cumulative, and that's surely better than animals getting sick and having to be treated with antibiotics and chemicals. So one of the projects I'm going to talk about quite a bit today is this polled project that I mentioned earlier, and this is the paper that came out in 2016, where basically Recombinetics introduced the allele from the polled allele from the polled gene into dairy breeds. So most dairy breeds are not naturally polled, Holsteins, for example, or Jerseys. And what it resulted in was these two bull calves that were born in April 2015 that did not grow horns naturally. And you might say, well, I know there's naturally occurring Holsteins and Jerseys that are polled because it's been introgressed many years ago from a non-horned breed, and so probably long back in their history there was maybe an allele came in from a beef breed. And part of the reason that you want to do this is because if you look at the genetic merit, and this is an economic index in the dairy industry, and if you look at the existing polled genetics, there's very few animals that have polled, and I should mention that not growing horns is dominant to growing horns, shown here. And you can see that almost all of the bulls in these two breeds are horned, in other words, little p. But more importantly, if you look at the genetic merit of the polled animals here, they're about $150 below the most elite animals in the dairy breed, which have the horned alleles. And so farmers have a natural tendency not to use these, and if they did use them, there's a very small genetic base from which to work it on. And so Macy, who did a master's with me, kind of modeled, well, if we were allowed to use genome editing in the dairy industry, how could we change the horned frequency? And shown here is if we don't do anything, this is what the horned allele frequency is going to be. In fact, it's almost 1 in the dairy industry. So there'll be basically no change. If we model that you have to use the existing PP sires, in other words, those polled sires, then what you would see is that you would over the course of 20 years, you would decrease the horned frequency to almost zero, which would be a good outcome. You could do it using existing PP sires, or the red here models if you could edit the top 1% of Holstein sires. And you see that you similarly got a good decrease here if you're allowed to use editing, but I think here is really the key point, and that is if you have to use the existing genetics, you would have a very, very big increase in inbreeding as a result of the fact that there's a very small little base of related animals that carry the polled trait in Holsteins and Jerseys. And so the inbreeding would go up quite a bit relative to the base scenario of not considering polled in breeding decisions. It goes up a little bit in editing, and this is basically the rate of genetic gain over time. And you can see if you have to use those inferior polled genetics that exist in the breeds, that you make less rate of gain relative to being able to use editing or just the conventional not considering polled. So those are some of the reasons that you might want to use this technology to introduce an allele into a breed without going through the kind of genetic bottleneck that might be associated with bringing it in from a different breed. And so, as I mentioned, not growing horns is dominant to growing horns, and anybody that's ever worked with polled animals knows that well. But interesting, if you look at the genetics, what happened here is the horned allele shown here is at the polled gene. What happens in nature was, many years ago, a 202 base pair allele replaced this 10 base pair allele. And so 10 base pairs got replaced by this duplicated 212 base pairs. And if you inherit this, you don't grow horns. And if you inherit a copy of this from mum and dad, because it's recessive, you will grow horns. And that's basically Mendelian genetics around polled. And so the bull that we had from recombinetics, we wanted to test whether or not, in fact, he was going to pass that trait on to his offspring. And so we knew from DNA sequencing that he was homozygous polled, and so he big P, big P. So he crossed him over a group of horned Herefords at UC Davis to produce some calves. And you can see them here, and as would be predicted, they did not grow horns. And basically, we did quite an extensive characterization of these offspring. So here's the original bull, so the gene-edited bull that's carrying the 2 copies of the polled allele, were crossed over 6 horned Herefords. Of course, they're going to be little p, because they're horned Herefords. And here's the 6 offspring, 1 female called Princess, number 1, and then 2 through 6 were males. All didn't grow horns. And then we also ran some controls along with this experiment. So we had some straight blood horned Herefords that just produced some horned Hereford offsprings. And then we had actually the dad of this bull's progenitor crossed over some horned Herefords to give us Hereford-Holstein crosses to be basically what we'd predict horned contemporaries for these guys, because this guy has the little p, so he'd be horned, and these all had little p, and so we got the 3 offspring there. And we've been following these calves that were born in 2017 for the last 2 years. And basically here you can see one of the Holstein-sired bulls alongside 2 of the bulls from the edited bull that didn't grow horns. They had little tiny scurs. But that's kind of the control group there. And similarly, here's Princess along with her Holstein-sired female control, and you can obviously see the phenotypic difference there. You may recognize Princess from an earlier talk I gave at BIF, where she was involved in being the cover of Wired magazine, and so Macy did a very nice job of getting her looking all pretty and ready for her cover girl shot. And you can see here, there she is on the cover of Wired, looking not at all like she actually looks in real life. But anyway, that's how the media tends to portray these things. But we basically phenotyped and characterized these animals for 2 years, looked at their health, looked at whether they grew horns or not, all of the different things you might do, and just published last week in Nature Biotech this paper, Genomic and Phenotypic Analysis of 6 Offspring of a Genome-Edited Hornless Bull. And basically, to cut a long story short, the calves had no horns and were otherwise healthy and phenotypically unremarkable. So no big surprise to cattle breeders here that that's the case. But what was unexpected and that caused quite a bit of media stir, and I want to take some time to explain it because I think it's important to understand what we did find, was that we found that one of the alleles that the bull was carrying also inherited the repair donor plasmid along with an additional copy of the Celtic or the polled allele. So they segregated in the offspring of this bull, and so some of the offspring inherited that plasmid and some didn't. So what do I mean inherited a plasmid? So let me try to walk you through this. It's a little bit molecular and I apologize. I do my best to keep it so it's comprehensible. So here's the naturally occurring horned allele, which basically is that 10 base pair piece that I showed you in the previous slide that was kind of simplified down. And basically what happened in nature was that orange section there was replaced by a repeat of this purple section. So this is the naturally occurring polled allele. This is the naturally occurring horned allele. So 2 copies of that you'll be horned, 1 or 2 copies of this you'll be polled. And so that is what the recombinetics was trying to accomplish, was to replace this with this. And the way that you do that is you provide the donor template, this 1.6 kb fragment, which is basically this sequence here. So here's the homology arms on either side, and this is the little piece that you want to replace this little piece. And then of course the editor comes in and cuts here. So that's basically what the intention was, and the intention would be at the end of the day that the bull would be carrying 2 copies of the wild type polled allele. But that's not what happened. So the bull was carrying 1 copy of the polled wild type allele, and that's just basically this sequence, which is the same as here. But additionally, there was not only what we were expecting, but there was the PCR. It was a backbone, plasmid backbone, an additional copy of this 1.6 kb repair template. And so how did that happen? Well, basically this is the plasmid that I mentioned earlier, and basically inserted in here was this the template, so the donor DNA, which is this 1.6 kb showing up here. And then this entire sequence is shown in yellow here. It also integrated into the DNA of the bull on one of the alleles. And so the genome edited bull carried the exact sequence of the wild type at this allele, so exactly as expected, and then also allele D, which is this guy, which included this additional piece of DNA, a 3.9 kb plasmid backbone, and then an additional PCR template, or 1.6 kb. And so in addition to what was expected, we had about a 5.5 kb insert on one of the 2 polled alleles. And just for perspective, what does that look like in the genome? So 5.5 kb is amongst the 3 billion base pairs that make up the bovine genome. So if you do the math, 5,500 base pairs over 3 billion, it's about 0.0002% of the bovine genome then has this addition. And of the offspring of this bull, 4, of course, inherited D, and 2, according to Mendel, inherited C. And so some of them got exactly what we were after, and so we could segregate this plasmid out. So what's the significance of this plasmid? Well, actually, it doesn't have any phenotypic effect on the offspring, and because this is bacterial sequences, we don't really expect it to have any big impact. It's just an insertion, a random deletion, or an insertion, sorry, that wasn't intended to be there but doesn't really have any phenotypic effect. And if you look at the genomic kind of comparisons across different breeds of cows, there's over 86.5 million genomic alterations when you compare different breeds of cows, and this is SNPs and insertions and deletions and all sorts of changes in the genome, which of course is the very basis of our selection programs, selecting for these basically mutations that give us characteristics that we want in our offspring. And so the other thing that was interesting and I think really important that we looked at was we sequenced all of the animals on this experiment, and it shows you this level of SNP variation that exists amongst the different breeds. And so we have 3 groups of animals here. This is the genome edited bull and his offspring. These are the horned Hereford bulls and their offspring, and this is the Holstein bull and his offspring. And what you see here is relative to the bovine reference genome, which you may or may not remember is horned Hereford, how many SNP variants we found in these animals relative to the bovine reference genome. And so this is the genome edited bull, and he had around about 6.4 million SNP variants different to the bovine reference genome. Here's his calves, which had about 6 million differences relative to the reference genome. And here are all the female mothers of these calves, which of course were horned Hereford. And so they have on average about 1 million less genetic variations relative to the reference genome than do their calves. But of course, they're still 5 million different because they're not the same animals as the sequenced animal of the bovine reference genome, the horned Hereford dominant. And you see that the dams in all of these experiments, which of course are horned Hereford, and in this case the bulls also were horned Hereford, that if you're in the pure blood horned Hereford group, all of the differences are around about 5 million. But if you go to an animal like a Holstein, then you get a whole additional magnitude of difference in terms of the sequence variation in the Holsteins, and that makes those calves quite different in terms of much higher number of SNP variants relative to the horned Hereford straight animals. And the reason I kind of emphasize this is that part of our evaluation of these animals was to look to see if there were any SNP changes that were what called off-target alterations. In other words, did we do anything we weren't expecting to do when we did the editing? And you have to kind of put that question in the context of this amazing genetic variation that exists within the bovine genomes. So that was a little bit wonky, and I hope I didn't lose anybody, but I guess the reason I think this is important is that the media got a hold of this story before the paper came out on August the 7th, and some of the headlines were quite kind of shocking and, you know, kind of a little bit fear-mongering kind of stuff. And basically that what we found was a plasmid insertion, and so I think some of these headlines overemphasize it a little bit, and basically they were reporting on a paper that was put into a thing called bioRxiv by the FDA, who were working with us on the evaluation of these animals and actually found the plasmid insertion. And this paper is again showing the template and the plasmid backbone, what an unedited cell line looks like. So this is homozygous horned, so that's the little P allele, and then this is what the bull looked like. So one, as expected, polled allele, and then again the one with the plasmid backbone and template. So that's basically the same data I just showed you, but that caused quite a bit of a media stir, and so I'll just explain that's what happened. So we've been working with these animals doing public outreach because I think we need to have a public discussion around the use of this technology, and we actually brought Princess, and this one with horns is called Cinderella, to a public outreach event and kind of explained polled and horned, and you can see science communication in action there. And we did a survey of a group of visitors to Picnic Day at UC Davis, and so Macy and I there are explaining what polled is and what horns are and what disc budding is and how we can introduce this allele. And we asked the audience before and after explaining that whether they would be supportive of the use of genome editing for this purpose. And just before I give you the answer to that question, one of the really interesting questions we asked was what percentage of animal products like milk, meat and eggs currently come from animals that have been produced using genetic engineering. And if you know anything about this field, you'll know there's only a single approved genetically engineered animal ever, the Aqua Advantage salmon, that's the fast-growing salmon, which has never actually yet been sold in the United States. And so the correct answer to this question is actually 0. And before we gave the presentation, we had the vast majority of our audience that thought greater than 75% came from animals that have been genetically engineered. I think they probably thought that we meant conventional selection, perhaps. But what was a little bit frustrating as a educator was we said very clearly in the presentation there was none. The answer was 0, and we did have some learning went on, but it's still a kind of a persistent number of people that just don't really want to take that message on board. So there's a perception that a lot of our animals are genetically engineered. But then if we look at the how do you feel about the use of genome editing to address an animal welfare concern, which was, in this case, we were using the example of polled. What I think is really interesting here, and this is a California audience, is 88% of the respondents at this event were either strongly or moderately supportive of using gene editing to address an animal welfare concern, i.e. polled. And that's quite different to some of the other survey data you'll see where 90% of people say they're opposed to quote unquote animal biotechnology. But if you explain why you're doing it, it does tend to change the perception a little bit. And I'm not the only one that found this. As a recent paper came out in PLOS ONE that basically asked another group of participants, this time in Europe, what their attitude was towards genetically modified polled cattle. And they found that many participants again reported positive attitudes. So that's a little bit encouraging. And so I guess to me, you know, editing is not going to replace conventional breeding. We're still going to be using genomic selection and have our breed associations and gather all our phenotypes and do our traditional genetic evaluations. Where it has a place is potentially as the cherry on top of a breeding program to introduce useful alleles without linkage drag, as in the case of polled, or potentially bring in useful genetic variation from other species. And so I think it has a place. And so as geneticists and people working in this field, I think we were quite excited that we might be able to use this technology in breeding. And we had news from the USDA in March 2018 that if you were not introducing novel DNA and you could have achieved the outcome that you got using conventional breeding, then the USDA was not going to treat those products as any differently to conventional breeding. In other words, you didn't have to go through quote unquote GMO regulations. And that came out from Secretary Sonny Perdue. And of course, the USDA is in charge of regulating genetically modified plants. Conversely, the FDA is in charge of regulating genetically modified animals. And in January of 2017, they came out with a guidance which considers that all intentional alterations in animals whose genomes have been edited are going to be regulated as animal drugs. And so overnight, these two bulls went from being polled Holsteins to being unapproved animal drugs. And if you look at the timeline for this kind of set of research, the animals were edited in 2014 and born in 2015. They moved to Davis in 2015. We collected semen and inseminated the cows in late 2016. Then this guidance came out whilst they were in utero. So overnight we suddenly had 6 unapproved animal calves or unapproved drugs on our hand. And we've phenotyped them for the last 2 years. We've collected meat from these 2 bulls, so they're no longer with us. We've sequenced and phenotyped all of the animals and shared that data with the FDA. The plasmid was detected in March. We've now had to kill all of the males that we basically got all the data we wanted from them, and we collected meat from them because the FDA wanted to look at meat composition. And Princess has been bred and we will get milk from her when she gives birth next year. But we were interested in knowing whether we'd be able to put these animals in the food supply. And this is the questions that we were asked, and they're kind of overwhelming, but this is the data that the FDA was looking for in order to make a determination as to whether animals could enter the food supply. And so obviously we had to say what we had altered and why we had altered it and how it was altered, and then we basically needed to compare the genetic sequence at the polled allele. And this is where it gets hairy if anybody knows how much work this involves. It's basically been 2 years of my lab's time. Doing whole genome sequencing data for the edited parental animals, the unedited cell control line, and at least 2 of the 6 offspring. we actually did all 6 because we got a grant from the USDA. And then this is the one that's really hard to address because these data should confirm the alteration, fair enough, as well as validate that there are no unintended modifications to the genome that impact animal safety. And that's a really hard question to answer. It's a little bit proving a negative. We also had to provide health records, nutritional composition analysis of edible muscle tissue from the animals, including the key nutrients. Any information to help the FDA in terms of a human food safety evaluation, and that information should be limited to those studies of edited animals at the Celtic allele, not from conventional breeding. In other words, we can't use conventional Angus, for example, and there is actually no other edited animals at the Celtic allele. And then for the heifer, we have to get the lactating milk. And this is for a trait of POLD. And so to me, that's not a trait that we would expect any problems with the milk and meat. It's not a trait that affects that. And to me, that's a little bit overkill. And so I just want to go through that we have this really strange regulatory system at the moment. So in America, these animals are drugs. In Australia, they basically say that if you use a donor template, that they're going to be considered GMOs. And so in Australia, because you used a donor template, you'll be regulated as a GMO, not conventional breeding. In the EU, if you use editing at all, you're going to be regulated as a GMO, whether there's any introduced DNA or not, because basically the EU said that if you're producing mutagens, then you're making a GMO. So all edits in the EU will be regulated as GMOs. And however, not if you use mutagenesis techniques that were in existence before 2001. So in other words, mutation breeding won't be covered. And so we have this situation at the moment that's almost unbelievable, where it's a bit of a toss-up where you are on the globe as to whether or not you're going to be edited as a GMO using editing. So the Latin American countries, particularly Argentina and Brazil, have taken a really strong lead in saying that you're going to not have additional regulations unless there's novel DNA sequence in the animals. And so if you haven't introduced novel DNA, then you're not going to be regulated. So the left-hand pathway knockouts won't be regulated, nor will alleles that already exist in that species. Whereas Australia, yes, because of the repair template. Canada, probably no, because it's not a novel trait. They regulate based on trait novelty. EU, yes. Japan, no. Again, they're like Latin America, saying if there's no exogenous template, you won't be regulated. And New Zealand, you'd be regulated because you used in vitro techniques, and the United States should be regulated as a new animal drug. There has been an order from the President in June of 2019 asking the regulatory agencies to basically update their approach and use their existing statutory authority as appropriate to exempt low-risk products of agricultural biotechnology from undue regulation. And that was issued back in June 2019. So in conclusion, gene editing offers an approach to precisely knock out undesirable traits or knock in desirable traits in food animal breeding programs. And it does open up new opportunities to basically bring in useful things like disease resistance. But at the moment, there's this patchwork of proposed regulatory approaches that will potentially be problematic for trade, even if, for example, you edit in Brazil, but you want to import into the US or EU, you know, you'd have to deal with the fact that the animals are edited. And really, to me, if these regulations aren't harmonized, then it's going to be really hard for animal breeders, especially in the United States, to use this technology. And I think the plant breeders in Europe are equally frustrated that this technology, which is kind of different to genetic engineering, is kind of falling into the same tired rhetoric associated with the whole GMO debate. So thank you, and I will pass control back to Darrh.
Thank you, Alison, very much. That's very informative. Does anybody have any questions for Alison? If you do, click in the chat and ask a question, or I guess you can go in the Q&A part. So let us know if you have any questions. I guess before I introduce Megan, Alison, I'll ask, and while you're answering it, I'll do the switch over. But do you, with light of where we are now, which seems so far away from this seeing the light of day, do you, in your most optimistic opinion, do you see us being able to usefully use gene editing in the anywhere close to near future?
Not with the current regulatory framework in the US. I do think it'll get used in other countries. I mentioned China has got a lot of scientists trained, and they're absolutely winning in the literature review around CRISPR papers. And so I think perhaps countries that have food insecurity that want to use this technology will go ahead and do it, not necessarily for products in trade, but to things like disease resistance. African swine fever is huge right now. If you could get an edit for that, I think that would be a really useful thing. So I'm not very optimistic in the US, to be honest, but I do think it will get used because it's such a valuable tool.
Okay, thank you. Now, and Alison, I think you're going to be able to stay on. If somebody has a question, they can still type it in during Megan's, and we can handle any questions at the end. Is that correct?
That is correct.
All right. If anybody has anything, please shoot them our way, and we'll move on now and introduce Megan. We have Dr. Megan Rolf, now at Kansas State University, and for those of you that may not know Megan, Megan is originally from Kansas. And has been involved with livestock her whole life. She got her undergraduate degree at Kansas State and then her master's and PhD from the University of Missouri. And she immediately out of graduate school went to work at Oklahoma State as an extension specialist. And since then, I think around 4 years ago, it's hard to keep up with time, Megan, she moved to Kansas State University, where she's taken on more of a research and teaching role at K-State. And whereas Alison has been winning the big awards, I consider Megan as one of our up-and-coming stars. And Megan is really all over the place, helping out with a lot that we do with, even though she gave up her extension role, she's still doing a tremendous amount of helping us with extension education as well. So Megan, I would like to welcome you and bring us up to date on the BIF guidelines, getting moved over to Wiki, if you would, please. And I guess I have to unshare first. There we go. Now you should be able to.
Perfect. Are we up and rolling?
Yes, you are.
Okay, perfect. Well, thank you, Darrh, for the incredibly kind introduction, and really pleased to be here today to tell you a little bit more about some changes that are happening with the BIF guidelines. In this case, the conversion of the guidelines to a wiki format. So even though I'm the one here giving the talk today, I want to pause and take a second to recognize the drafting committee, because I've done a very small proportion of the work compared to all of these folks. And they would include our host today, Darrh Bullock, as well as Bruce Golden, Warren Snelling, and Matt Spangler. And then we've recently been fortunate enough to recruit Merlin Nielsen to work on editing the guidelines as we finish up different sections and work towards a full launch of those. Okay, so just like our previous guidelines, the target audience for the new guidelines really isn't any different. So still a valuable resource for breed association staff and leadership, for producers themselves that may have questions about, for example, how to record data for specific traits. Useful tool for industry professionals, and then of course students and academics, as well as professionals within the extension system. So nothing really different about the target, but what is different really is the format. And so one of the big questions that always arises when thinking about why move to a wiki format is really some of the advantages that go along with that. And the first and one of the biggest advantages in my mind is the fact that instead of the BIF guidelines being a PDF static document, with the conversion to a wiki format, what we really have the opportunity to do is make these guidelines a living document. And the advantage of that is that we don't have as much risk of those guidelines becoming stale over time, because we can really continuously update this document and address science as it happens. So when technology evolves or when we have new scientific developments, we can go in and make the appropriate sections and edits to the wiki and really have that be a real-time document that addresses things in a more timely manner. One of the other advantages is that it can really be maintained by the stakeholders. So as the stakeholders are using the guidelines, things can be identified that may need some work. So maybe we identify something that needs to be updated, or someone is working on some new research and says, hey, that'd be a really good fit for the data processing section. You know, we can update all of those things due to stakeholder demand. As well, I think it's a little bit easier to use. So it does curate information in one location, and that was true arguably for the old guidelines as well. But I think what is true of the wiki format is that it's much easier to search and find information within the wiki format. And we'll actually go through and look at some options for that later on today in the presentation. And finally, we have instant delivery to all devices. So it really doesn't matter if you're using Android or your computer or whatever that may be. You're able to access the wiki and utilize the information within it. So related to the fact that the wiki is more conversational, I think the whole herd reporting section that I have here up on the screen is a good example of that. So one of the things that the wiki format lets us do is really set up some templates for content, and then we can go through and really have some conversations surrounding some of these subjects and have a lot of interconnectivity between all of the different pages within the guidelines. And I think that creates more of a conversational setup for all of the information. I mentioned that it was more searchable, and here's an example. So even straight from the home page, if you don't want to go through any of the sections specifically, for example, if you wanted to really find out some information on selection index, all you really need to do is type selection index into the search box at the top of the page, press enter, and it's going to bring you to the page specifically on selection index. So you can read all about selection index, and you can even be linked directly to the scientific resources that were used to develop that page. So there again, increasing some interconnectivity between different pieces of information. So if you really want to jump to a specific page, in addition to searching, maybe you don't necessarily know exactly what page you want to jump to, you can always visit the table of contents. So the table of contents can be accessed on the left-hand side of the screen under the BIF logo. So if you click on table of contents, it will bring up this page that you see on the screen here. And it basically has an alphabetical listing of all of the pages that exist within the wiki. So maybe you didn't know exactly what you were searching for, but then when you see it, you realize that you really wanted some information on body condition scoring. So you could pull up the body condition scoring page, get a great table that outlines each of the different scores along with color pictures, and some information on how to actually do the scoring, complete with the original scientific references used to develop that page. Finally, I've mentioned the scientific references a couple of times, but I wanted to point out this page on accuracy because I think it's a great example of how we can embed those references into the guidelines. So in the previous guidelines, one of the downsides of that format was that for some of the sections that had been developed a long time ago in the past, maybe the authors had retired or were no longer involved with BIF or not on the board, not involved in guidelines revisions. And so occasionally there were questions about where the scientific resources were housed that originally were used to develop those guidelines. And sometimes if that person is not around to answer those questions, we might not necessarily know. So one of the things that the new guidelines does to solve that is it really embeds the scientific references within the text. So you can see here on the accuracy page towards the bottom, we have 3 scientific references here. They're directly referenced in the text here. And then from that, we can link directly to the actual citations for those and then link to the papers themselves. So there's no longer any question of where that information came from. It can easily be accessed and retrieved, even if the author decides not to continue to update the page as an example. There's one other bit in the wiki format that I think is useful. In the old guidelines, there was a very large appendix that had a lot of information in it. And a lot of that information is incorporated into the current guidelines, even in the wiki format. But we do have a page that sets up a reference section of a lot of resources that could be useful for different groups of individuals interested in the guidelines. So, for example, on the essential reading page, there's a section for industry professionals that includes things like the NBCEC beef sire selection manual and some other foundational information targeted towards industry professionals. If you're a grad student, you're either really going to like this page or really hate this page because it has a really large section of really foundational animal breeding papers in the scientific literature that'd be great for new graduate students in animal breeding, and even for those of us that have been around for a while and haven't read those papers in a little while. Those things are really outlined quite well in the reference section. So I wanted to take a second and just maybe switch over to the wiki format and flip through a few pages and talk about some of these things in real time. So I'm going to go ahead and try and get that pulled up here. Okay, so hopefully, and Darrh, tell me if it's not showing up correctly, but you should be seeing the landing page for the Guidelines for Uniform Beef Improvement Programs. So from the front page of the guidelines, you can see there's a link to email Jane Parrish, who's the executive director. There's also a link to the 9th edition of the BIF guidelines. But this is basically the place that you'll start when you move into the wiki format here. From there, if you really weren't sure what BIF was, you can click on About BIF and take a look at the different pieces of information about BIF. You can move specifically to specific sections by clicking those links at the top. One of the interesting features of the wiki is that it's actually really useful and easy to link to outside pages. So for example, if we want to know who the member organizations are that are part of BIF, we can click on that link within the text, and it'll redirect us to BIF's page that has all the entire membership list. Or if we were reading down towards the bottom and we really wanted to know a little bit more information about the genetic prediction workshop, we can pull that page up and go right to viewing that information as well. So if we go back to the home page, from there, we've already clicked on one of them, but you can see the main headers to the page are really at the top. And so if we click on another one of these, let's just go ahead and go to selection and mating. So from there, you can see the conversational tone of this information. So maybe we're reading through here and we decide that we want to know a little bit more about, let's see, what do we want to pick here? Maybe we want to know something specifically about Cavanese maternal EPDs. So we could click on that. It would take us to the page that discusses that information in terms of calving difficulty, look at the scoring system and all of that information and how that information is utilized in genetic prediction. You could also try and click on, whoops, didn't want to click on that one. There should be one in here on heifer selection. But let's just try pure and straight breeding. So if we wanted to get some additional information on that, we can see some information here, as well as link to the appropriate sections of the sire selection manual as an example. or learn a little bit more about inbreeding by visiting some fact sheets. So maybe within this, we want to know a little bit more about selection. Here's where we can access the information on replacement heifers. So maybe we want to know about replacement heifer selection. You can click on that. It'll bring us to some more information as we're reading through there. Maybe we want to know a little bit more about, for example, what we might be interested in selecting for, or what our options were for selection on growth and milk production. So that would bring us to a list of traits that are categorized here based on growth, reproduction, carcass traits, management or convenience traits, etc. So maybe within that, I know that my breed has scrotal circumference EPD, so I'm going to click on that and find out a little bit more information on how scrotal circumference is incorporated into genetic evaluation. And again, you can see the embedded references at the bottom of the page. From there, let's go back to the main page. Maybe we're interested in genetic evaluation. So from here, we read through, maybe we want to learn a little bit more about single-step genomic BLUP versus single-step hybrid marker effects model. So we could click on both of those two pages and read down through that information. See, we have lots of great figures and a lot of references here if we want to go to the literature and check that out. The same for the marker effects model here. So we have all that information, and we have the information in literature that we might want to go and look a little bit more deeply at. Maybe we don't want to get that technical, and instead we really want to learn more about what economically relevant traits are. So we've got a little bit of information here, and again, we're able to link to a fact sheet through eBEEF.org. And if this gets pulled up here, we can see that is written by Matt Spangler, so we can pull up the PDF version of that, and then we can actually look at some tables here that discuss what the economically relevant traits are and what the indicators are and how all of that works in the context of genetic evaluation. So if we go back to the guidelines here, the other piece I want to point out is also on the left-hand bar. So these are the author resources. So if you're reading through the guidelines and you decide, hey, I'm really working on a lot of cool research in X, and X really isn't in the guidelines, you can always contact one of the section editors and discuss adding a section to the guidelines relative to that. And here you can see the resources that are available to authors of the guidelines. So with that, I'm going to head back to the PowerPoint, hopefully. And put in one last plug that if you have any information on a specific trait or something relative to data collection and processing, or any good idea that you have for the guidelines, please feel free to contact the section leaders. And of course, if you're reading through the guidelines and you happen to think that we need to update one of them, that's something you can also let the section editors know as well, because that contributes towards the wiki being a living document that really evolves as science progresses. And that, I'd be happy to answer any questions that anybody might have about the wiki.
All right, thank you very much, Megan. Does anyone have any questions for Megan? I'm going to go ahead and pull up next week's agenda while you guys can think about that. If you have any questions for Megan or for Allison, please type them in. I know we beg for questions every week, and it's just not something we get a whole lot of. But if you have anything, please let us know. We'll try and take care of them. So while you guys are thinking or typing your questions in, I'm going to go ahead and give you the agenda for next week. Session 4 is next Wednesday, of course, at noon Central Time, and Dr. Weber is going to be your host next week. We're going to be having a talk on the genetics of meat quality and Bos indicus cattle by Dr. Raluca Matescu from the University of Florida, and then also going to have from the University of Florida, Dr. Matt Hersom is going to give you guys the information on BIF for next year, and we invite you all to please join us for BIF next year. And so if you want some more information on that, Matt is going to bring us up to date on that. All right, we have a question, Megan. Approximately when do you think the BIF Wiki site will be mostly complete?
Okay. Well, I'm going to interpret that or answer that as mostly a little bit of a trick question. So mostly because the guidelines will never necessarily be complete because it's an ever-evolving document. But in terms of the majority of the content that we're planning on having in there right now, to actually answer your question and not avoid it, is that I think we're pretty close to having a lot of that information complete. And part of the process with Dr. Nielsen going through the guidelines will be to identify things that we need to continue to add to the guidelines or work on a little bit more, and then we will continue to work on those sections and bolstering those sections that he identifies. Hopefully that'll be done within a couple of months after we get started, but it will kind of continue to evolve over time, so I don't know that we'll necessarily ever say that we're done, but it's certainly complete enough at this point that if someone wants to go in and start taking a look through it, they're certainly more than welcome to do that. And if you notice anything missing, feel free to want to let one of the section editors know.
I guess, Michelle, I would follow up to your question by saying I would guess that next at the BIF in Florida is probably when the board will probably give the final vote of a complete switchover, wouldn't you think, Megan? I mean, that's the next time the board will be together. So I would think that that's probably, I think we'll be done before then. And like Megan said, I think it's a very usable document now, and you can go ahead and start using it now. But I think that couple of months till it's, you know, almost complete, and then official probably move over and adoption would be at next year's BIF.
Yeah, thanks, Darrh. I should have mentioned that. That's, yeah, I think that's a fair statement.
Any other questions for either of the speakers? I really appreciate you guys joining us today, both Megan and Allison, and thanks for your expertise on these. And I guess if there are no other questions, we will end the session and look for you guys back next week. Be sure that Joe Pascal and Steve Hammock and those guys know that we're going to be talking about the Indicus cattle next week, so get them back on here. All right, folks, look forward to seeing you next week, and thanks for joining us. So long.